Hello and welcome to our second Bio40 Pre-Class lecture. Today we're going to explore Mendelian traits in humans. Let's start by quickly reviewing Mendel's discoveries. He showed inheritance is particulate, with units he termed genes. There's no blending. Most cells have two copies of each gene, but gametes only have one. There can be different alleles or forms of each gene, and they segregate randomly. Alleles are dominant or recessive, and a homozygous dominant individual and heterozygous individual will display the same phenotype despite having different genotypes. Different genes will sort independently. Let's move away from peas for a while. No offense to peas, they're certainly like a top 20 vegetable, but we would certainly have very little interest in pea genetics unless the knowledge applied across species. And generally as humans, we most want to know how these rules apply to our own biology. We couldn't actually do experiments like Mendel's using humans. We can't control crosses, there are no self-crosses, the number of progeny from each couple is too small for proper statistical analysis, getting to an F2 generation would take several decades. Additionally, it's hard to find good traits that are controlled by single genes. Most human phenotypes involve complex interactions of multiple genes. However, there are some traits controlled by single genes in humans, and the best studied ones are alleles that cause disease. This table shows a few of the most common genetic diseases caused by single genes. There are dominant and recessive examples, and they behave just like the dominant and recessive traits Mendel observed in peas. In humans, genetic traits are best followed with pedigrees, a form of family tree. Here we see different symbols for male and female individuals. Mating lines connect parents, and lines of descent connect to their offspring. When a couple has multiple children, the siblings are connected by a sibship line. Each generation is labeled with a Roman numeral, and each individual within a generation is given a number. This allows you to specifically reference each person in the pedigree. So this individual is 1.1. She is 1.2. The children here are 2.1 and 2.2. The coloring of the symbol indicates who has the disease. Empty symbols are unaffected individuals, while filled symbols show they have the condition. Occasionally you'll see a partially filled symbol indicating a carrier, an unaffected heterozygous individual in the case of recessive conditions. For several progeny, you'll sometimes see them represented by a number inside a symbol in order to save space. Individuals who have died are indicated with the slash, and a double mating line is a consanguineous or incestuous mating. Let's start by looking at cystic fibrosis. This is the most common lethal genetic disorder among people of European descent, affecting about 1 in 2,000 newborns. Patients have a variety of respiratory and digestive ailments. Median survival age is now above 50, and luckily this is a number that I frequently get to revise upwards. In the 1980s, this number was below 20. Here are a couple pedigrees of families carrying the allele for cystic fibrosis. This information is enough to tell us the allele is recessive. If a trait is absent in both parents but emerges in the child, it must be recessive, and both parents must be carriers. Additionally, we can conclude each unaffected sibling has a 2/3 chance of being a carrier. Here's another pedigree that's a little more complicated. After several generations without an occurrence, the disease suddenly appears in individual 6.4. So where did the allele originate? One possibility is that one of the parents in generation 1 was a carrier. They passed the allele to both children who are also carriers. The allele continued through the generations, but no one married another carrier and had a child homozygous for the allele. That is, until these distant cousins had children, and one ended up inheriting the recessive allele from each parent. Another possibility is that it didn't originate in generation 1 at all, and two people who married into the family both happen to have the allele. For common conditions, this is possible, but the rarer the disease is, the more likely the first scenario is, since it only requires one individual to introduce the allele into the family instead of two. This illustrates why inbred families have a higher incidence of rare genetic diseases. Let's assume the disease is very rare and the allele originated with individual 1.1. We can assess the probabilities that various unaffected individuals are carriers. 2.3 must be a carrier since the allele had to have passed through her to reach her great-great-granddaughter. As a father of the affected individual, 5.1 must certainly be a carrier. 3.1 married into the family where the allele is already present. She's no more likely to be a carrier than anyone in the general population. Over in this branch, 5.2 must be a carrier because his child has the condition. And finally, individual 7.2 has a 2/3 chance of being a carrier since both her parents are carriers, but she is unaffected. When it comes to recessive traits, most alleles that cause serious diseases are rare. These diseases will often skip generations. The parents will typically be unaffected carriers. Inbred families have a much higher incidence of homozygosity because the allele is rare amongst outsiders. When two heterozygous carriers mate, 1/4 of their offspring should be affected, but due to small sample sizes, you may see large variations on this. Finally, in the circumstance that two affected individuals mate, all of their offspring will be affected. Now let's look at another condition called Huntington disease. It is very rare, ranging in incidence from 1 in 10,000 to 1 in 1 million individuals, depending on the population. It is a severe neurological condition that doesn't show up until adulthood, typically between the ages of 35 and 50. It leads to death typically 10 to 15 years after onset. Here's a pedigree for Huntington disease. Since the affected children always have an affected parent, i.e. no skip generations, this indicates a dominant trait. In theory, a pedigree like this could be the result of a recessive trait, but that would require that unaffected individuals 1.2, 2.2, 2.4, 3.5, and 4.2 all be carriers that married into the family, and for a rare genetic disease like this, that would be exceedingly unlikely. So assuming the disease is dominant, we can again determine the genotype of many individuals, or at least the probabilities of them having certain genotypes. Individual 1.1 may be heterozygous or homozygous for the dominant allele. Homozygosity for rare dominant alleles is very uncommon though. In fact, for many dominant diseases, homozygosity is embryonic lethal and such individuals are never born. This is not the case for Huntington disease though. And given that 1.1 passed the allele to both her children, and we have no info on her parents, it is a possibility. Individual 3.1 is homozygous recessive. If she had any other genotype, she'd be affected. Now let's look at the 14 children of this consanguineous mating. The parents, 3.6 and 4.6, are both affected, but both have an unaffected parent themselves, meaning they are both heterozygous. Like any monohybrid cross, we can say there is a 1/4 chance that any given child will be homozygous recessive, and thus unaffected. So individuals with dominant traits will always have a parent with the trait. If you see just one example on a pedigree of a person with the trait without an affected parent, you can conclude the trait is not dominant. Affected individuals should transmit the trait to half of their kids, but again, small sample sizes means that there can be big deviations. It is possible to have an unaffected child of two affected parents, and indeed about 1/4 of their offspring should be unaffected. However, if homozygosity is not viable, that number will be more like 1/3. Those small sample sizes will make it very difficult to tell the difference. And of course, one big issue with humans that didn't come up in pea plants is sex. Spoilers for next week, but genes are present on chromosomes. And in humans, males have an X and a Y chromosome, while females have 2 X chromosomes. We say males are hemizygous for genes in the X chromosome. They only have one copy, so they will have the phenotype corresponding to whatever allele they have, dominant or recessive. Here's a pedigree for hemophilia, a disease common in European royalty that affects people's ability to form blood clots. Because it skips generations, we can tell it's recessive. That there are female carriers but only males are affected is a strong sign the condition is X-linked. In general, X-linked traits will appear much more often in males than females. The only way females can have the trait is if the father is affected and the mother is a carrier. Because fathers pass on their X chromosomes to daughters and Y chromosomes to sons, affected fathers will never pass on the trait to sons, but all their daughters will be carriers. Hypophosphatemia is a condition where there's insufficient phosphate ions in the blood. In this pedigree, you can see that every affected individual has an affected parent, suggesting the allele is dominant. It is likely X-linked because the condition never passed from father to son, but is inherited by every daughter of an affected father. With affected mothers, the trait is passed to half her children regardless of sex. This all means that X-linked dominant traits are more common in females than males, However, very few such conditions exist. Even more rare are Y-linked traits, but they would be easy to identify because they never appear in women and are transmitted to every son of an affected father. Another odd pattern you'll find in pedigrees comes from mitochondrial inheritance. Mitochondria are organelles that contain their own genes separate from the chromosomes. When the sperm and egg unite, the sperm does not transfer its mitochondria, so all mitochondria, and therefore all mitochondrial genes, come from the mother. Pedigrees of mitochondrial disease are distinctive in that the traits are never passed from father to child, but are passed from mother to all children. That'll do it for this lecture. Next time, we build upon Mendel's laws with some extra complexities.